Hi, everybody. Thank you so much for having me. I've really enjoyed the symposium so far, and I'm super excited to talk about some of the research that we've been doing over the past year, looking at biodiversity and ecosystem functioning from a below ground perspective. So as we are aware, right, we are losing species globally at an unprecedented rate due to anthropogenic forces, right? And this loss is projected to only get worse in the future. So an important question to ask then is how this species loss is going to affect the remaining species, the ecosystems they used to reside in, and the global cycles that we rely on. And through decades of research, we know that as we lose biodiversity at any trophic level, we lose ecosystem functioning. And ecosystem functions are broadly the biotic and abiotic processes within an ecosystem that residents, including us, depend on, right? So this graph on the left shows the decrease in ecosystem function that we see as we lose species. And we see that this is a pretty dramatic decline. So as well established as this relationship is, we are still unsure of the mechanistic link between biodiversity and ecosystem functioning, or what we call BEF. Meaning we know this relationship exists, but we have yet to uncover the drivers of this relationship, which is essential for maintaining ecosystem functioning in the face of species loss. And so how do we address this gap? One way may be taking a trait-based approach. So rather than looking at the species level, we can quantify functional traits of each species. And functional traits often reflect the species strategies for acquiring resources and interacting with other species. So what are species doing in an ecosystem and how do they do it? And so this approach hasn't offered us the insight we'd hope to find, right? This graph shows here the number of traits included in a study, along with the percent of this BEF relationship that's been explained by those traits. And often it's less than 10%, showing us that it's not explaining a large portion of this relationship that we'd hope that it could. So what are we missing? And it's important to note here that the vast majority of functional traits measured in plant communities are strictly above ground traits. So we are failing to capture the below ground information, which is vital to understand how plants uptake their resources and compete below ground, right? So it may be time to take a new, more comprehensive approach to this trait-based method. Using Texas grasslands as a study system, Texas grasslands offer an extremely unique perspective because of the extreme and ever-changing climate that we're so aware of. And this allows us to study the effects of species loss on ecosystem function in the context of global change. So in this experiment, we used a 12 species pool of native Texas species. And in each of these plots here, we varied the number as well as which species are in each community. So we varied plot richness from 1 to 12 with the addition of a water treatment. So half of these plots were water to replicate the anticipated precipitation change regime, anticipated change in precipitation that we expect to see, right? And so this photo shows part of our experiment. The total thing is 144 plots. And it also shows these insect nets that we have. For time, I will not go into them, but I'm happy to talk about them during questions if we have time. But to understand how below ground functional traits may inform this BEF relationship, I needed to dig up some plants. And I dug up a lot. So for each species, I collected whole root samples from the watered and ambient plots from the 1 species and 12 species plots. And for each system, they were cleaned and photographed as you see here. And then these photographs were processed into a black and white photo. And then these photos were analyzed using fancy computer software. And this quantified the below ground functional traits. So all in all, it was just over 400 individuals. It only took me 3 months to get all these digitized. So that was really fun. But these trait values were then used to calculate a metric of functional diversity, which essentially just tells us how different the plants are within each community. And our primary measure of ecosystem function in the system is productivity, which is just the dry biomass of each plot during the peak growing season. And so with that, with this massive undertaking, we found some really interesting results that we're really excited about. So first, we do see a significant increase in productivity as we increase species plot richness. So this relationship holds true in Texas ecosystems, that as we increase diversity, we increase ecosystem function. However, we see that this fails to explain a lot of this variation. So we're still not fully explaining this relationship. And so maybe this below ground functional diversity metric may offer some insight. But it doesn't. It doesn't have any correlation with productivity, and that might indicate that below ground traits are not as informative as I expected. However, if you start to look at these traits individually, we start to see some interesting trends. So this graph shows the average specific root length within each community, and specific root length, or SRL, reflects the amount of carbon invested towards below ground resource uptake and competition. And so we see that that productivity increases the more resources are devoted to below ground. And this is a significant relationship. And so it indicates that plant communities are more productive when more resources are put towards below ground, and that this variability at the community level is really important. But what about at the individual level? One critical assumption I've made within this below ground functional diversity metric is that those values remain constant across all treatments. So I decided to test this assumption and look at trait plasticity, or how these trait values change across treatments. And I found that in fact, species are incredibly plastic in their root traits. So again, using SRL as an example, we see that these individual species are changing their specific root length values across these ambient and watered plots. And this is a significant relationship. So we're seeing some dramatic responses in these plant communities as this water availability changes, right? Which makes sense. Plants need to respond to their environment. But do they respond to the differences in changes in their plant community? And yes, right, across these 1 and 12 species plots, we also see dramatic and significant changes in this specific root length as well, indicating that plants not only respond to abiotic, but biotic changes as well. And so the big takeaway from all of this is that species level values are not always indicative or reflective of every population, and we need to be able to take this into account. And so in order to effectively use this trait-based approach to understand this BEF relationship, understanding trait plasticity is key. And so by furthering this understanding, we'll first be able to achieve our primary goal of better predicting this BEF relationship using more realistic trait values that offer this mechanistic insight that we really need in terms of maintaining ecosystem function in the face of species loss. Additionally, we can understand how plants may respond to global change, and how plants will be able to adapt and acclimate to these climate changes given their plasticity levels. And lastly, at a more fundamental level, we can begin to understand how these various stressors influence the plant community as a whole. And so thank you so much for listening. I want to thank my wonderful collaborators and mentors, along with the labs I'm a part of, for all of their amazing insight, and the whole slew of undergrads that we had giving up their time and effort to make this whole thing possible. I also want to acknowledge my funding sources, the wonderful Native Plant Society of Texas, as well as the Stengel Wire Endowment at the Department of Integrative Biology. And thank you again for listening.
Sara, thank you so much.